Wonderful. Thank you. So let me just give you some of the updates about the research that we have been doing over the other end of the world in Australia. So like how Dylan mentioned, that our priority is to integrate the perspective and the needs of the families. And this is where, in collaboration with KIF1A.org, we thought about how we can create a research program which is very much focused upon putting the projects in line with what the families really need. So we had this first Australian research program that's focusing on CAND and what we now call it as kinetic. So kinetic really means as KIF1A investigations into neurobiology, expression, transport, innovation, and care. So it's all about three main pillars. So the pillar one is about understanding the biology behind the disorder, like what is happening at the cellular level, just so that we can get to know about the pathways which get us perturbed. And for that, what we are doing is we're using different model systems. One of the main things is going to be iPSCs. I'm so glad that Jane and Marcus has touched upon what the iPSCs are, but more likely these are the cells which is derived from the CAN individuals, and that they can be formed into neurons in organoids. And at the same time, we have got some more simpler model systems which could be used for the high throughput drug screening, which I have touched upon in the previous conferences. Pillar two, it's about, again, just to focus on how we can enhance the early diagnosis for CAND and how we can accelerate the treatment options, including the high throughput drug screening and the gene therapy. Pillar three, that is something very important. That's about empowering the CAN families. Why do we want to do that? We want to co-design the tools with the families so that the support services which could be developed for them, it's keeping, again, their needs, their priorities in mind. So as a part of this pillar, one of the things that we really want to do is to establish the national network. In that particular network, we aim to have researchers, clinicians. We want to have other diverse experts as well. For example, the qualitative researchers, health economists, and all of the different people who could bring in their expertise on the table and say that this is a combined thing that we can do for our community to be, what do we call as a clinical trial readiness. So for the purpose of today's talk, I'm only going to talk about pillar one and pillar three. So when we last met in New York 2 years ago, there was a lot of families talking about how the vision is really impacting the life, the quality of life, and the progress in KIF1A children. This is where we thought about what we could do in the space, and the quotes that you are seeing on the slides I have pulled from the KIF1A.org website. It just tells about again the importance of studying CAN and how the clinicians and the researchers should put some effort, more effort into understanding what are the different mechanisms because of which the vision changes is happening in the CAN children. With that in mind, we wanted to, again, look at, to complement with all the clinical studies which is happening, that is there a way that we can, again, model vision in the lab and can study it a little better? So again, just drawn from the findings, the clinical findings that's been reported and collected as part of the KIF1A study, led by, again, Dr. Wendy Chung and Dr. Alaya Abdul Hakim. So as part of this study, it was kind of reported that almost all of the CAN children, they have got a range of visual problems, but one of the most common one which is coming up is the optic nerve atrophy, that nearly 95% of the children, they have got their optic nerve getting changed because of the KIF1A mutations. So with that in mind, normally how the optic nerve is measured clinically, there are two things to it. One is the structure of the optic nerve. The second one is about the function. So when we talk about the structure, it's a technique called OCT, optical coherence tomography, which talks about how thick or thin the optic nerve is. And then the second technique, which is on the board you might see, is the VEP. That's that's the visual evoked potential. So what the visual evoked potential does, it basically looks at the functionality of the eye brain network. So when we call it an eye brain network, Dr. Mary Whitman yesterday explained really well about how these two integrate, but this is just a schematic about how our eyes, they act as a camera. So they will take a picture, and that information will then be sent to the brain, and the pathway they follow is the optic nerve. So in a way that our eyes and brain, they are connected structurally as well as functionally. It's a very famous saying in the ophthalmology world that the back part of the eye, what we call as a retina, it's like a window to the brain. We can peek through what's happening in the brain as some of the signs and changes which is happening in the back of the eye. So that could be a really powerful tool if we could know a bit more about what's happening, what are the changes which is happening in the KIF1A children, that can they be indicator of what's happening in the brain. Now with the retina, it's a very complex but very organized structure. Like this is a schematic that I've pulled together in which you would see different layers of the cells. Now it might get a bit confusing. So in a way, if I could simplify it, just imagine there is a light which is coming from the bottom. So when the light comes from the bottom, it first hit the retinal pigmented epithelial layer. So this is the epithelial layer which, if there is too much light coming into the eyes, that's going to be full of glare. We don't want to damage the cells which is underneath. So this layer absorbs any of the extra light and then only filters through the light which is really needed to interpret the signal. Then comes the photoreceptor cells. So the photoreceptor cells would convert the what we call as a light energy into the electrical energy, and that's way it kind of keep traveling up, up, and up until it reaches the top layer where it'll find the retinal ganglion cells. Now, the retinal ganglion cells, what we call as the RGCs, these are the first type of cells which is formed when the retina is still forming. So quite important in a way that they are the neurons. So these are, they will have their projection. So they will take the message which is coming from the below part of the eye, but their axons or their extensions would come together and they will form the top yellow layer, which is called the RNFL, the retinal nerve fiber layer. It's basically the optic nerve, but it's still in the eye. So it'll take a 90-degree turn and come out of the eye and form the optic nerve, which then gets connected to the brain. So bit complicated, but I think the key message that I want to give it here is that the optic nerve is mostly formed from the RGCs, the extensions from the cells, which is called RGCs. Now, if you cut the optic nerve, you would see that there is fascicles, and if you keep pulling these the nerve fibers from the fascicles bit by bit, you would end up with the minimum layer, which is the axonal layer. Now, given the KIF1A is a molecular motor protein, so there is the hypothesis, we believe that KIF1A has got a role to play in terms of the axonal transportation, so it might be having some role into maintaining the optic nerve health as well. But before we could dive into the lab experiment straight away, we first asked this question that, is KIF1A expressed in retina at all? So there is a very cool thing about all the modern technologies that all the assays which is done as a part of the global studies, they get transferred into databases. So we can always go in and check it's something like, hey Siri, I want to check about what is the level of KIF1A in this particular cell line or organ. So we asked this question in terms of what is the expression of KIF1A in the, first of all, in the cells of the retina. So on your right, you can see that there is a graph which shows about KIF1A expression that's from the mouse optic, the mouse retina, and the lines along the x-axis, it just shows the different types of the cells. But what you could do is you could just focus one from the second last. I believe it's the RGCs. So I think this was a good confidence for us to see that yes, the KIF1A is expressed in retina into different cell types, including the RGCs. And more typically, if you see on the graph on the left, you could see that this is the data of the KIF1A expression from the human human cells in terms of across the development phase. So you would see that there is a graph. The KIF1A expression starts off quite high, but then it starts to drop down during the first trimester of pregnancy, and afterwards it gets stabilized a little bit. So it again shows that KIF1A has some kind of a role which is early on in the development, but then it got stabilized over the course of the development. With that in mind, we first wanted to study the optic nerve atrophy using the mouse model system that the JAX lab has developed. And this work would not have been possible without the collaboration with Dr. Kat Lutz and Dr. Marcus Terry, in which Marcus and his team, they were really helpful in a way that they gave us access to the eyes, which is joined with the optic nerve from 3 different mouse models that they have developed with the leg dragger, P305L and T99M variants. So they took the eyes out with the optic nerve and shipped it all over to us in Australia, and where my team then looked at what's happening in terms of the structure and in terms of like what are the changes which these optic nerve and the retina in the mouse model is happening. So so I'll start off with some of the study results. These are very pilot findings. That's from the optic nerve. So on the left, the top, you can see there is an optic nerve, which is really round in shape. It's pink in color. It's quite big, and you might not see because of the lighting, but there is like blue dots in between. That's basically the cells in the nerve. Now, the top panel is the wild type, but if you look at below, that's the optic nerve which is taken from the heterozygous P305L mutant mice. What you could see, one of the most prominent thing which among the two, it's basically their size. So on the top, the wild type has got a bit bigger size, whereas the P305L het mice had a relatively shorter or the smaller optic nerve. The other thing which we noticed, which might not be very clear, is that the intensity or the number of the blue dots in the bottom picture, they are relatively much higher as compared to the top picture. Now, we don't know at this stage whether, so this is what we call as a cell infiltration. So it looks like a lot of cells are coming in to support the optic nerve. Now, this could be just to kind of supporting cells to, if there is some kind of a changes which is going on into the optic nerve, that a more supporting cells wants to come in, they want to join and help preserve the degradation. Or it could be that the sign or the markers of the inflammation into the optic nerve. So we don't know at this stage, but again, like if we think about the quantitative analysis, so on the right-hand side, we can see that the P305 het mice optic nerve was 1/3 the size of the wild-type healthy nerve. Then we did look at what's happening at the retina. So I've put the schematic on the left just as a reference so that it's easier to understand. On the right-hand side, you would see 2 pictures put across to each other. So on your on the left, it's the wild type retina, and on the right, it's the P305L heterozygous retina. So you would see that again, we could see all these layers of the retina quite clearly with this H&E staining. The striking difference of all the layers is from the top. At the top with the RGCs, the cells which was making the optic nerve, they are housed. So in the wild type, you can see this layer is a bit thicker, but in case of the P305L het mice, that thickness is reduced. So again, because this is something new that we are studying in the lab, so in order to be very confident that the changes that we are seeing is real, we do have to find out different ways of looking into it. So we have looked through the cross-section of the optic nerve, we have looked through the retinal layers that again kind of supports that there is some kind of a changes which is happening into the layer in which the cells that make the optic nerve are housed. Moving further, so that's great. We have got 2 pieces of evidence, but the next piece of evidence that we want to collect together is specifically looking at the RGCs in kind of a very deep lens. And this is where we look at some of the markers which is very specific for the RGCs or the retinal ganglion cells. So one of the very specific marker, I know I'm bringing a lot of sciencey words here, it's called the BRN3A. So BRN3A, it's you can think about this as a protein which is mostly expressed in the RGCs. So in this panel here on the left, if you see the green, forget about what's happening at the bottom. But if you look at the top layer, I don't have my pointer onto it, but you will see that it's like bright green speckles which comes on just the top layer. These are the RGCs. So what we want to achieve by doing a particular staining of the RGCs is that we want to calculate the number of these cells on those top layers and then compare, is their number really changing between the wild type mice and the P305L het mice? Again, this is going to give us a bit of an indication that there is it could be a cellular loss at the RGCs level, which is then traveling back and then having some changes at the optic nerve level. So we've got some findings which is showing some key findings that yes, there is some changes in the optic nerve from the mouse model, but these mice, if I've not mentioned before, they are aged mice. They are nearly about an year and a half of age, so to be precise, 16 months old. So that's a very long time to kind of see these changes coming up. But at the same time, like how Marcus Terry was saying, that we only have a luxury of 3 different mouse models at the moment. So if you want to extend it beyond these variants, if you want to find some complementary model systems, human iPSCs is the way to go. So because these are the cells which has been directly derived from the can individuals. And if we've got these iPSCs, and if we can force them to make the cell types of our interest where we want to do, if we want to investigate the role of the KIF1A. So this is the same thing we kept in mind when we were making the brain organoids. So in terms of, in this particular study, before I could link it up with the vision study, I just want to take you through what we have found in the brain organoids. So when I say brain organoids, it's kind of like a mini brains. So we start off with the iPSC cells. We either force them to make the 2-dimensional neurons, or we try to differentiate them into 3-dimensional organoids. So again, on the left, on the top, you can see this is an organoid, which is what we call as the isogenic control, or we can think about this is as the healthy a mini brain from a healthy individual's iPSCs. So this is, again, at the bottom, it's the CAN individual's organoid with the variant R203S. As we can see in terms of these organoids, when they are about 2 months old, they've got this very nice tight band at the top. But whereas if you see at the bottom, this tight band is basically relatively very thicker. So what it signifies is, again, there is a changes in the structure at the organoid level when these organoids are about 2 months of age. Now, at the same time, if you see next to it, you would see the green speckles. So it, again, the intensity of the light might not make the justice, but you would see that this green marker is basically the indication of the neuroinflammation in the brain. It's a marker of the astrocytes. So in case of the isogenic control, we see that the GFA expression was again localized to that band at the top, whereas in case of the organoids that we derived from R203S variants, there are some points in which there is a high intensity of these GFP markers, GFAP markers. So again, it's a kind of a indication that there is a stress which is going on into the organoids which have been derived from the R203S variant. So structurally, they are different. And again, at the point of the markers of the neurodegeneration or the markers of the stress, that's again different. But then we next wanted to ask about, are they different at their electrical output endpoint as well? That when we say that, it's about like how our brain, in like how Jean was explaining that there is a technique in which there is a special dishes, what we call as the MIA plates, MEA. So what it does is that every time the brain organoid is going to fire, it's going to capture that signal and it's going to compare that whether the rate of that firing is same between the wild type or the healthy or versus the CAN individual's organoid. So if you can see into the graph here, so the 3 parameters, which is at the top headings, so this is just a different way of looking into it. And you would see that in case of the, what color it is, pink. So pink is the CAN organoids. So in case of the CAN organoids with the R203S variants, the firing pattern is different as compared to the wild type. So again, it shows that there is a defect, what we call as difference between the wild type and the R203S variant at the structural level, at the inflammation level, and as well as at the electrical activity level. So with that in mind, we thought about, that's great, like we could recapitulate some of the features for CAN into the brain organoids. What if we now combine the brain organoids with the eye organoids? Are we going to see any kind of changes into the optic nerve because of the KIF1A? So this work was inspired by the work of Associate Professor Anay Gonzalez Cordero from Children's Medical Research Institute back in Australia, where they have done something similar for their disease model, in which they have got the brain organoids and the eye organoids. They are both growing next to each other, and you would see there is a thin red line which is kind of connecting the two. So that's what they call as the optic nerve-like projections. So this red line represents the RGCs growing from the eye and connecting with the brain. So this is not a mature optic nerve yet, because it's going to take time for it to mature, but there is still a very good positive sign that there is a possibility of growing the brain with the eye that's been connected with the optic nerve in an organoid level. So with that in mind, we thought, let's give it a try with the KIF1A. So are we able to achieve something similar? So what we did was that before we could jump on to looking at what happens in the CAN patient organoid, we wanted to look at just with the healthy wild type, not we say healthy, but the isogenic controls. And in that, you can see that at the bottom of this figure, that represents the brain organoid. And at the top, it's the eye organoid. So within the eye organoid, you would see this black dot. So that black dot is basically the retinal pigmented epithelial layer that I was talking about. The reason it is black is because it captures excess light. So that gives us an indication that yes, we are able to see something which naturally happens in the retina, that this organoid is actually having that supporting cells coming up. But what is even more interesting, and that's where we want to focus on, is the retina. Do we see the retina or not? So if you see that the white translucent band, which is coming across the end of the eye, starting from the RPE, that's the layer which is ultimately going to make the retina. So it's quite promising that we have been able to kind of recapitulate some of the features which is in the retina into this organoid model. Now, this is about 2 months old organoid, but what happens is that the optic nerve-like projection starts to come at about 2.5 months. So we are quite excited to see that if we leave these organoids up for a little bit more longer and then do sectioning and then have a look at the optic nerve projections, we are quite positive that we are going to see something similar into the canned organoids as well. The next step would be, again, what we want to do is we want to look at the development of this optic nerve in the CAN organoids and then compare it with the isogenic control organoids just to see what is the rate. Is there a kind of a rate of a difference of the development of these optic nerve-like projections between the two different genotypes, healthy versus the variant? So again, once we start working onto these model systems, we have to look really carefully about, and we have to be confident that what we are seeing is actually real. So the only thing, because they look very different, you would say that this doesn't look like a brain and this doesn't look like an eye. So we have to go at the molecular level and then have those investigations and have those evidence to prove that yes, what we're seeing is actually responding or corresponding to the right organ system. So these are the slices what we have stained for some of the proteins which should be expressing in the brain part and something which should be only be expressing in the eye part. So on the left two images, you would see the staining with something called SOX2, TBR1, TBR2. So you don't have to remember what they are, but I think in a nutshell, these are the things which should only be present in brain. And if you could see the staining, that they are actually represented in the bottom part, what we call as a brain organoid. In the last image, if you see, there is a pink staining, and that's the pink is the marker for the CRX. Now, CRX basically stains the photoreceptor cells. So it's a very, again, what we call in general, this is a very eye-specific molecular marker. And if you could see that this is only hanging at the top one, which we call as the eye organoid. But what is more interesting is that KIF1A is there. That's what we really want to study, right? So the KIF1A is there in the brain part, which is as the green color, and you would see it as in the eye organoids as well at the top. But the more important thing is that if you see that band at the top that I was talking about, and this is the band which will make the retina. So the KIF1A is present in that particular band, and we were quite excited to see that there is some evidence that yes, we can make these organoids which is connected to each other, and that the KIF1A is present at the brain level, and it is present at the level of the retina too. So with this in mind, so what's the next step? What are the next plans? So our first plan is about, we do know that the optic nerve atrophy starts to, basically we could see that it has already happened at about 16 months of age of a mice, but when did it started? So what was the course of the optic nerve atrophy in the mice model from the beginning to the 16 months? This is where we want to longitudinally take some time points and look at the health of the optic nerve at that particular time points in the P305L het mice, and then compare it to the wild type, something similar what we have already done in the H mice, because this would really give us a bit of a timeline, like how it is developing, what changes are occurring, and at what time point. So that why do we want to do it? Because again, if this is something which is kind of degenerating, or it is kind of slowly changing in the shape and the size, we want to capture a time point when the changes are the earliest, so that we can stop those degeneration or the changes quite early in the timeline. So that's one thing. The another thing that we do want to do is we want to take these mice, and then we want to look at the functionality of the eye-brain network that we were talking about. So if you remember, these are the two studies, the OCT and the VEP studies, which is mostly done in the CAN individuals. What we want to do is we want to do something similar on the mice as well, and then compare with each other. Like, now, this is again needed, the reason because we've got a mice which is showing some kind of optic nerve atrophy, but we want to be confident that the changes that we are seeing with the mice actually correlates with what's happening in the CAN individuals. And again, I think here the theme of the conference is again coming about more participation. So again, if we are going to compare the findings from the mice to the human, to the CAN individuals, this is where we are going to get it related with the koala study. So more we know about what's happening in the individuals at that level, more it gives us opportunity to compare, like, are these model systems actually working? Is it kind of a true copy and the true representation of what's happening in the CAN individuals? The next aim would be something that we want to again extend for the organoid studies. So we've got some success with the isogenic controls in which we have been able to make the brain and eye organoids connected with the optic nerve-like projections. We want to extend this study to the CAN organoids, and we want to look at what's happening across the different time points. And one of the other aims that we really want to focus on is that we want to start off with the canned individual iPSCs, and we want to force it to make the RGCs. So if you again remember, RGCs are the cells which basically would make the optic nerve later. So once these RGCs gets differentiated, we want to first characterize how the RGCs from the canned individuals, they are different from the isogenic controls, and then really want to see what are the changes which is happening. And then what we want to do is we want to treat them with the small molecules that we have identified as part of the high throughput drug screening, and then look at like whether any of these molecules actually can rescue the phenotype differences between the healthy versus the canned individual RGCs. With that in mind, I'm going to shift a little bit of a gears towards pillar number 3. So if you again remember, pillar 3 is about empowering CAN community. So how, again, here I just want to focus back again on the priorities and the needs of the KIF1A families. So over the years, while working with the KIF1A.org, we have identified that there are certain areas that we need some more study on, more research on, and one of which was about understanding the speech and language in KIF1A children. And this is where we had this amazing collaboration with Professor Angela Morgan and Lottie Morrison, who really just came on board, who took us under their wings and then did this study. Lottie presented beautiful findings yesterday. So I just want to say thank you. Thank you so much for doing the speech study. Great. So, and we know that collectively, the rare disorders, they have got such a high economic burden. So when we talk about the dollar amounts, that's quite high, like it's in trillions of dollars. But what we are missing on is what is the dollar figure in CAN? Like what is the health economy burden while caring for KIF1A children overall? So that link is missing. And this is something that we really want to focus on next year, in which we have got these collaborations with, again, the experts in the field, just who could really let us study about overall impact of CAND when it comes to the dollar figures. I think it's quite important because when we go to the policymakers, when we go to the pharmaceutical companies or anything, it really helps us find our pitch that why do we want to do it. And even now we look at the cost of care, cost of hospitalization, and there is different cost in terms of the healthcare, but we don't appreciate so much about cost of taking time off from work, cost of caring for the child. So this is all very small, I won't call it small, but it's very big factors. So we really want to study this in more detail and then have those figures ready so that it's more published, it's more like evident, it's more visible, just to again show the effect of the CAN at different levels. And this is something, again, this was very close to my heart. I always wanted to do this. So that's about the lived experience study. So it's a very qualitative study. So we are researchers, scientists. So we look at numbers, we look at molecular pathways a lot more. But then I got this, there was a lunch, and then I got in touch with some qualitative researchers who look at the lived experience. And I was like, oh yeah, we really want to do something for CAN as well. Would you like to join? And they were amazing. So they came on board, they said, yes, let's do that. We had an undergrad student, Kara Mivadale, so who took it as a part of her undergrad studies, and here we started on the qualitative lived experience. Now, when we say the lived experience study is important, why do we want that? Again, all of you, I mean, each one of you are doing so much about making the community aware about what CAN is, what is the impact of CAN on the community, and it is really empowering in the way that everybody is getting to know more about getting a more feel of CAND. But at the same time, I think it is so important for researchers like myself, Mike, Jane in the audience, for us to know that what is really important for the families. And this is where I think this is where it brings a different perspective for us researchers to have our projects planned as well. It is so in the qualitative world, like all the blogs, all the videos, all the experiences that you have been sharing, it comes as a gray literature. So that's really helped the policymakers to have that holistic view about CAND. But when it comes on the on what we call as the evidence, it gets even more impactful if you've got some academic literature published. And this is where, when we started to have the plan for the study for the qualitative research, we wanted to look at, is there any academic literature which has been published already, which we can take as the backbone? We can take it as a reference and then create a hypothesis and then look at the study, what we really want to answer the questions on. There is not much about the academic literature about CAND except for these two studies. What do we call as the first being the opinion letter that's been published last year by a group in India who talked about the impact of the physiotherapy in CAND and what is the impact on the children and impact on the families, which is great. And at the same time, it was quite nice to see that there was one study, a one-book chapter in the book called Shared Struggles. If anyone of you could go to that book, I mean, that's really beautifully written book. And specifically chapter number 27, which says, "Can I give you advice?" And that's about the diagnostic journey of our very own Gia Grace, who is not here with us at the moment. So I again want to take a moment just to remember her and just to say that you are loved. And at the same time, I want to say thank you to her lovely parents as well, just to kind of come up and really share and really contribute as that finding about like what it was like for them when they were going through the diagnostic journey, when they were meeting the healthcare professionals, when they were not getting the answers, until they found one person from the healthcare that really was able to connect at that level and then really could actually give them that piece of advice. So yeah, thank you. But then otherwise, if we look at, if we go back onto seeing that how we can really contribute to change of the policies, change of some like bigger pictures, which is like as researchers, which is beyond our control, but this is something that we can contribute, is about, again, there is a significant gap on the published peer-reviewed publications, which talk about the lived experience of the CAND families in a fashion that how the qualitative researchers would like to report for them to finally change some of the policies and to, again, to co-design, co-develop some of the support services which the families really need. So that's where Kara and I, we had this amazing time with the Australian and New Zealand families. So we had one-on-one Zoom interviews with many families where I would say we laughed together, we cried together, and it was really one of the beautiful conversations I have ever had. And the reason being because we touched upon some of very personal aspects of the life, in which what was their journey like before the diagnosis, how was it during the diagnosis, and then up until to future perspectives. But the highlight was about what was the enablers, what were the complications they had, and what were some of the challenges they faced, so that again we could find out these common themes which is coming out, that we can then bring it out, publish them, and have them ready for the community to say, in case if they're going for, I'm not sure how it works here, but back in Australia we have this national agency called National Disability Insurance Scheme. So what we call as NDIS. Now NDIS is a national body which basically supports a lot of therapies for the children with additional needs, including CAND. So be it occupational therapy or speech therapy, physiotherapy, and everything. But what happens is that every year the funding needs to be renewed. And when the renewal of the funding comes, it's really tricky. I mean, I have this experience personally, but it's not easy to convince the person on the other side of the call to say, this is what is canned, this is what my child is going through, and this is why we need so intensive speech therapy or so intensive occupational therapy. So I think this is where this particular study would come in picture, so that once it is published, they don't have to go through all over again every time of the year when they have to really go for the funding renewals. They can simply take that paper with them and show it to them and say, look, this is what we really mean and this is what we want to say. Again, having said that, the experience for all the families is unique. It's very different. The way it touches everyone is going to be very different. But again, I think the idea of the study is to find out those common codes, those common themes which comes out, which I believe it's going to be very powerful. So again, like I said, we had one-on-one interview with the families for about an hour, and we touched upon the questions about the diagnosis, about the future perspectives, how's the everyday life, like what it is being living with CAND. And then again, I think through this conference and even engaging with the Australian and KIF1A families, there is something common which is again coming out. That is what is the lived experience of the siblings, like how the siblings wants to be heard, and then how is their experience of caring for their brothers and sisters and even parents is impacting them, and how this is really of strength. So I'm coming back to Leila's talk the other day. So again, I think this is something that I'm going to integrate back into the study and see if we could do something extension in which we particularly ask and speak with siblings to know about their perspectives as well. Having said that, coming back to the study, we had the interviews. They were on Zoom. They were recorded. So this is very fancy way, or like of just recording, and that there is softwares which could actually transcribe those interviews, so you will have it in the Word format. Now, as a lab researchers, this was a very different experience that we have learned, very big learning curve. But then just to make sure that we are actually capturing the right information, every interview had to be screened word by word so that we are confident that what we're going to analyze is what actually the parents said. After the transcription, what do we do is that actually Kara did that with all the qualitative researchers. So they'd look at these coding. They would go through the interview, they'd look for the specific codes, and they make certain themes of those interviews. So it's a quite nice, quite informative, and quite different process, I would say. But then that's the code, and that's the themes which comes out. And being a qualitative research, it's the codes that's powerful, because that's coming from the families. It has emotions, it has frustration, it has every bit of compassion, passion, everything in it. And we want to capture that. And this is why it is so different from the quantitative study. Instead of being the numbers, it's about the depth, and it's about their experience of how caring for a child, and then even living with CAN, how it means. So when I say that, so again, like I said, it was a pilot project. We wanted to start off with the Australian and the New Zealand families, and it was amazing to see 17 participants enrolled and took part in this particular study, and they were the representative for 19 GAN individuals. So that nearly about 70% participation. So again, I want to take a moment here and say a big, big thank you to all the Australian and New Zealand families to, again, believe in the project and take part. Thank you so much. So it's quite small text, so I won't be able to kind of read it from off here, but the theme which was under the healthcare, so some of the things which I think we have been hearing a lot about, that also came out as part of this interview process. That was about dismissal by certain healthcare professionals when the parents, they raised their concern. They talked about how early diagnosis was not possible and how it would really change the trajectory of their CAN children if it was provided early. And they also, sorry, Dylan, can you help me read what's the second and the third points?
More practical information.
Oh, yes. So one of the parents had said that they really didn't want to know what KIF1A is, what is the biology. We don't want to know. We just wanted to know what is the practical information. Can you connect us with other families around the world? Because there is more meaningful conversation that can happen between carers and the parents. So that was their perspective, and I quite liked it. Oh yes, that's the very big thing which is coming out from the Australia and the New Zealand families, and we see that, we hear that, and we believe that because they have had this experience with a lot of different healthcare professionals, and I think it's getting better with time because there is more awareness, there is more understanding what CAN really mean. But a lot of health professionals, they don't know about CAN, what it is, so they really wanted to kind of have that. Let's say if we talk about Australia, they really want to have that network within different states, so that there is one neurologist, one ophthalmologist in, let's say, Melbourne. There would be somebody in the Sydney, somebody over at Perth, and all across Australia, and they would know what CAN is, and then they can talk to each other. But it is making much easier for the family, so that they can stay locally and they are still very well cared for. And again, I think it matters in a way because we want to have we want to care for them longitudinally. We want to kind of care for them over the time. So this is where, again, having one expert, which is quite tricky, I would say, but having that one expert is really important for the study and then for the families as well. And this is where I think I kind of quite get excited when Dr. Wendy Chung mentioned about the international network, which is getting formed here, so that families will still have the opportunity to talk to Dr. Wendy Chung, so that because she knows about what CAN is, but at the same time, if they really want to go and speak with a neurologist locally in Australia, they would know, okay, this is my go-to person. I can just go, I can show what's happening in my body, and then they will be able to kind of guide us. So this was a kind of a very consistent, powerful theme, which was coming out of the qualitative study that we did. I might not be able to read all the quotes, and I don't want to do any injustice by saying something else. And these slides will be published, and this will be getting recorded. So please take your time and then have a read about like what the families have to say. We have got this wealth of data with us, but in terms of what we really wanted to present as part of the study, we have just taken a common quote from a particular theme, and then we have included it here. But like how the researchers, they were quite amazed with how engaged the network was when we were asking the questions. And even the qualitative researchers, they said that we have got this wealth of information which could be analyzed at different levels to make an impact even more higher. And then we wanted to look at the impact on the families. Oh, great. Yes. So I'll just probably pull up the slide as well so that you can read along the codes. Financial burden. Like I mentioned, we have got NDIS in Australia, but it was still quite surprising to know that the family on average can spend up to 1,500 Australian dollars a week to care for the children outside of what support they were being given by the government. So that's massive.
Individuals with CAN may have increased sensitivity to sound and light, which can be stressful for parents.
So stress, yeah, this was again coming out quite a common theme. Like it's really the families will have happy times together, but then again, there are moments of that extreme stress, and it could be as little as what one of the parents said here, that even the rolling of a sticky tape from another room, it can really make a child not liking it, not being happy, that how it actually creates then stress on the parents, which is yeah, it just shows again like the impact on the families on the daily life.
Vision issues are challenging and require parents to adjust their parenting approach.
So again, like I think as a parents, we always have to change approach, but then the impact even more significant when there is issues about vision, when there is issues about movement, and which we have been hearing about a lot from yesterday's talk. So again, it again at a level, it does create that stress, and it does create that they have to change the parenting style. I was speaking with Leila's mom before, like how we find ourselves at the crossroad when like both the children need our attention, and then we have to really keep changing our parenting style so that we can make them feel heard and included at the same time. So again, this is quite significant, which was coming out as a theme of the qualitative research.
And relevant to our dance party on Thursday, music is a powerful coping strategy for families.
Oh, yes, that's right. Almost all of the families, they were saying music is the go-to thing. Like if we have exhausted everything, there is nothing much left to say, turn on the music, and they would have some better moments following that, especially for the children.
Slowing down degeneration of the disease is a high priority.
Wonderful. Thanks, Dylan. So like I mentioned, we did ask the families about what are their research priorities. And the two main things, again, which is in line with what we have been hearing throughout this weekend, that's about the strategies to slow down the degeneration, and then coping with the vision, understanding vision a little bit more, so that, again, there is ways to support the vision, there is ways to treat the vision moving forward.
And families value research focused on understanding vision issues in individuals with CAN.
Mm-hmm. That's, again, in line with the same thing, which is, again, I think it's quite... First of all, the families, they are sharing about, from their lived experience, that what is it like when the child really wants to kind of go in, enjoy the free space, but then how vision really limits about their progression, like what they have to learn, but then at the same time, it really inhibits about how it can interfere with their speech, or with their falls, and with all the different aspects of life. So it was quite clear, and at least now this is qualitatively through this conference. I guess if there was a qualitative researcher here in the conference, they would be amazed with how much data they have already captured. I mean, the survey that Dr. Wendy Chung did at the initial, this is quite nice as well, because that's the information that they have already kind of gathered throughout the conference. So having said that, these were the initial, the pilot results, but we do want to expand globally. We do want to have this chance to speak with the parents, including yourself and the others online, and just see if we can find the same patterns of themes, and to get more perspectives of the different families across the globe. So if you're interested, if you can please scan the QR code, it's only going to ask you 3 questions. Whether you're interested, yes and no, feel free to say no. But if it is yes, then please put down your name and the email address, and then we make sure we reach out to you. We'll give you more information so that you can make an informed decision whether you still want to continue, and then we can have the study and the interviews with all of you. We'd love to engage with you and know your perspective as well. I think this is a very nice segue to introduce you all with KIF1A Australia. So I didn't make these slides. These slides are prepared by Ms. Janet Harris, who is the founder, who's a co-founder, I would say, for KIF1A Australia, KIF1A.au. By the way, I just missed that this koala, it's a limelight. Everybody is liking this rainbow koala. So it's a great representation. So Janet and David couldn't be here at this conference, so they asked if I can present these slides on their behalf. So at last year, Janet and David Harris, they came together, and then they made KIF1A.au, KIF1A Australia. Janet and David are the parents to Summer Harris, who is a 4-year-old, a beautiful child, and she has got CAN. They live in Queensland. And again, the mission of KIF1A Australia is the extension of KIF1A.org. That's about raising awareness, having advocacy about CAN, and at the same time, accelerate research so that we can find, again, more treatments, more cures with time. And again, I just want to take a moment back again, just hold on here, because they are doing a lot about raising funds, about doing all the fundraising, just to support the critical research at Murdoch Children Research Institute. So I just want to say thank you to them. And at the same time, they've got the fundraising in terms of, they won't leave any of the any of the podcast interviews or anything of that sort. And along with what Gary Boyer, Catherine, Edward, and the family they have been doing for so long to connect the Australian families, and I think they have just joined the team, and it's going to be even more bigger impact when all the families could come together and they just go in one direction about raising awareness and talking about CAN. So they have mentioned some of the more highlights. Again, Dylan, please give me an indication when my time is up. So they do talk about CAN in different avenues, like, for example, the Link magazine. That's, again, Australia's national disability magazine. And that's a magazine for the children with additional needs. And KIF1A was highlighted for the first time. And in fact, the reason I'm saying that, when I went for the physiotherapy appointment for the physiotherapy appointment, and there was a physiotherapist who talked about, have you looked at, do you work on this particular gene? I've read the story in the Link magazine. So again, it just shows that awareness, that impact, with what creates even a small thing can have a bigger stories. And for the next couple of slides, it's just kind of a snapshots for the KIF1A Australia Day that they hosted back in April this year. So with that, I just want to say thank you. And I've got these beautiful pictures of our lovely children from Australia and New Zealand. Just wanted to say a big thank you for them. And thank you so much for inviting and allowing me to share the research updates from Australia. Thank you so much.